Hi, this is the Organisation Reverse the Classroom video. Now, the actual topic itself is quite huge, and what it does, it goes through cellular organisation, the digestive system, there's a required practical food testing, enzymes, digestive enzymes, effects of temperature, pH, then it gets onto the heart, then we talk about coronary heart disease, all about the blood, the lungs, health issues, non-communicable diseases, and then we also do quite a lot on plants as well. Plant systems, transpiration, translocation, and then active transport at the end. Okay, now starting off with hierarchy in living organisms. What it could be is they could actually ask you what each one of these individual things is. So if we start off with cells, so what a cell is the basic building blocks of all living organisms. Then what they do is they just get bigger. It goes to tissues, organs, organ systems, organism. So what you've got to do is just got to remember that order. So what a tissue is a group of cells with a similar function. An organ, I like to think of it as like a group of tissues with a similar function. An organ system is a group of organs working together with a similar function. So that's organs working together. And an organism is a group of organ systems working together. So what there is a pattern where it's a group of all the things previous in the list working together to get to perform a function. Examples, there's loads of different cells. Okay, we've done about cells before. Muscle cells, nerve cells, sperm cells, all those different kind of things. The muscle example is good because then what you do is you get muscle tissue, and then lots and lots of tissues working together. Could be the heart. The heart is the major organ in the circulatory system, and then organism, you can have any plant or animal. Now, enzymes. Okay, it's quite a big sort of topic in this. Now, what the whole point in an enzyme is, particularly the digestive enzymes, is to speed up a reaction. Digestive enzymes, what they do is they take large molecules and they turn them into small molecules so they can be absorbed. Okay, that's the whole point in digestive enzymes, is to take big proteins, for example, turn them into smaller, which is then your amino acids, then they can be absorbed. And the lock and key model works by you have a specific enzyme and you have a specific substrate. The substrate goes into the enzyme, so it's kind of the enzyme here is the lock. The substrate is the key. The key goes into the lock, and then what you do is you get the enzyme substrate complex. Then what happens is the enzyme does whatever it's got to do, and out pops the products. Another important thing to remember, that there is called the active, can't write very well there, active site, and that is a specific shape. Now, enzyme activity, there's two main things that we talk about to do with enzyme activity. They can be affected by temperature and pH, so how acidic or alkaline something is. So this then at the bottom here is then the graph to do with temperature, and you can see here the optimum temperature, which is that blue line, is about there. That's the optimum temperature, which funny enough is about 37 degrees C, which is normal body temperature. But what you'll notice is the graph kind of goes up gradually like that. It gets to the optimum, and then what it does, it then denatures. So denature, that's all one word. Okay, and what that does, it means breakdown. Right, it doesn't really mean breakdown. What it means is the active site changes shape. Okay, but what it's doing, it's only really denaturing that direction. With pH, your optimum pH here is about 7, which funny enough is the pH of the human body, near enough. And what happens if it gets too acidic, it denatures, and if it gets too alkaline, it denatures. Right, this is one of the two required practicals in the organisation topic. It's the enzymes required practical. Now, I'm not going to go through all the details of it, because what you need to do is you need to go and find the video that goes through all the aspects of the actual practical itself. Right, but the whole point in it is to look at enzymes, amylase, effect on starch. Okay, this then just shows a bit of the practical equipment that's going on, and this then shows the results, which is kind of based on pH. Right, but again, I'll just say it, go and find the video to do with enzymes required practical and study it in detail, because there's a pretty good chance it could come up on an exam. Now the digestive system. What I'm just going to do is I'm just going to start off with the structure, and then we'll talk about each individual part. So the whole point in the digestive system is take out food, take out whatever you need out of it, and then get rid of the sort of waste products. So we start with the mouth. So I'm just going to write here, mouth. Okay, and I'm not going to write out the full things here. Then it goes down the esophagus, which is this tube here, and it goes into the stomach. Then when it's gone into the stomach, which is then here, it then goes through the stomach, and when it pops out, right, there's 3 other organs that kind of have an involvement in the digestive process, but the food doesn't actually go through them. They are the liver, the gallbladder, and the pancreas. So food doesn't actually pass through them, but these 3 things add things to it, to the digestive process. Once it's gone from the stomach, it goes into the small intestine, large intestine, not fitting it on very well. Then often it will say rectum, so rectum is then just around about here, and then it goes to the anus. Okay, so that is then the actual process of the digestive system, all the different organs that are then involved. Now this is then what each individual part does. So salivary glands produce amylase, right? And what amylase does is it breaks down carbohydrates. Okay, and that is then produced in the saliva. The saliva also lubricates. Okay, and what that then does, it then allows you to sort of swallow a little bit easier. It then goes from the salivary glands, it goes into the gullet. Now the gullet's a terrible word, right? You need to use the word esophagus. Right, and what esophagus is basically a tube that takes the food from the mouth down to the stomach. And it kind of uses a process called peristalsis, which is where the actual food is squeezed down. Hence why you can stand on your head and drink a pint of water, because the esophagus actually squeezes the food down. Then we get into the stomach. The stomach has got 3 roles. Number 1 is it churns the food. Okay, and what it does is it churns the food, mixing it with the acid, and there's also an enzyme in there, pepsin, which breaks down proteins. And it's very unusual as an enzyme because it works at pH 2. Okay, so it breaks down proteins into amino acids. Number 2 there, it actually says stomach produces the protease pepsin. Third one, and this is the one that I think you should always write down in the exam if you're asked for a function of the stomach. Number 3, it produces stomach acid, which kills bacteria. Okay, so what I'm going to write down here is sterilize. Sterilize. Okay, what it does, it sterilizes the food. So when you have your burger, there's tons of bacteria on it, there's no denying it. Right, and what happens is the food goes into the stomach and the hydrochloric acid sterilizes it, kills all the bacteria, so you don't get ill. Right, so once you've been into the stomach here, what happens is it comes out of the stomach, and then those are those 3 organs that I talked about before. All right, so firstly, the liver. What that does, it produces bile. Now, bile has 2 jobs. It neutralizes the stomach acid, so that is an alkali, right? Because if you imagine if you had stomach acid pH 2 and it went all the way through your digestive system, I'm sure it can't be particularly very pleasant coming out the other end as pH 2. So bile neutralizes it. That then gets it to the right pH for enzymes to work in the small intestine. The second job is it produces bile, which emulsifies fat. And what that does, it means it breaks it down into smaller globules. If you think about when you're doing washing up and you've got like fat on the top of the water, put washing up liquid in it, and all the fat globules break down. That is emulsify, so it breaks it down. Okay, so that's the liver. It produces bile, and bile's got two jobs. The gallbladder, that stores the bile. All right, so it's kind of just like a little bag next to the liver, and it stores it ready for putting it into the digestive system. The other one is pancreas. Now the pancreas produces amylase, proteases, and lipase, right, which breaks down carbs, proteins, and fats. Okay, I like putting lipids because that's kind of like the technical name for it. Carbohydrates are broken down into glucose. Protease are broken down into amino acids. And fats and lipids are broken down into fatty acids and glycerol. Okay, so that's what those three enzymes do, and those three enzymes are then put into the small intestine to continue the process of digestion. Then, once we've gone past those, and again I'll reiterate the fact that food doesn't actually pass through those three, the liver, the gallbladder and the pancreas, all it does is they add things into it. Then we get to the small intestine. The whole point in the small intestine is to absorb everything that has then been digested into the blood. All right, now if you think about the 7 food groups, so what it will do is it'll absorb the digested protein, so it'll absorb the amino acids, it'll then absorb the glucose, it'll then absorb the fatty acids and glycerol, it'll absorb minerals, and it'll absorb vitamins. Okay, so then they've been broken down into small things so that what they can do is they can be absorbed easier, because your small intestine couldn't absorb full-size proteins because they're too big. They also produces amylase, protease and lipase to help finish off the digestive system. Okay, now if you think about it, it then goes from the small intestine into the large intestine. In the large intestine, theoretically, and obviously this isn't 100% true because the digestive system isn't 100% perfect, what you've got left as food groups is you've got fibre and you've got water. And the job of the large intestine is to absorb water. So in the small intestine it's very runny, and the large intestine, what it does, it thickens up the stools ready for evacuation. And finally what you do is you get to the rectum and the anus. The rectum stores the feces until the exit through the anus. Now, even though I've talked about it already a little bit, right, this is then the enzymes in the digestive system. Carbohydrates, like amylase, they're made in the salivary gland, the pancreas and the small intestine. And what they do is they break down carbohydrates to simple sugars, particularly glucose. Okay, so what they're doing is they're taking big molecules, making them into small molecules so they can be easier digested. Proteases aid in the stomach, which is pepsin, and the pancreas break down proteins into amino acids. Amino acids. Lipases, right? Lipases, lipids, it's all to do with fats, right? Made in the pancreas, work in the small intestine, and they break down lipids to fat. I always say fatty acids the other way around. I say fatty acids, that's double T, and glycerol. Okay, those two are then small enough then to be absorbed. Now, even though I've got a fourth one here, right, bile in the enzyme list, bile's not an enzyme, right, but what it does, it helps the digestive process. Okay, right, we've already talked about it. What it does is it emulsifies lipids to increase the surface area for the enzymes to work on. Now, this is then the second practical. Now, I'm not going to go through the details. Look up the video to get all the details of it. But there are basically 4 tests. The iodine test for starch, the Benedict's test for sugar. They like that one because there's a lot of good colour changes. the test for proteins, which is the Biuret test, and the test for lipids, right? There's a few of them, all right? What you could do is you can do ethanol and water. You've then got rub on brown paper, and you've also then got something called Sudan 3. But I'll just say it again, look up the other video to get the full details. But obviously the methods are here anyway. Now the heart. Okay, now obviously what you're going to get in your exam is you're going to get a heart diagram that looks like that. Might be a bit simpler, well, it will be black and white. But what you've just got to do is you've got to kind of work out the order that blood kind of flows through the heart and the circulatory system. Now, this diagram on the right hand side here is ridiculously simple, right? But what it does, it kind of shows the pattern. So what I'm going to do here is I'm going to start with the right atrium. And again, notice that the right and the left are on the wrong way. And what I'm going to do is I'm going to kind of start off with the right atrium, and I'm going to find the right atrium over here, which is there. So what blood does, it collects in the right atrium. Then what it does, it goes through a valve. And I'm just going to keep changing the colours. There's a valve here. Okay, these white strands here is all kind of part of the valve. So blood goes from the right atrium through the valve into this area here, which is the right ventricle. The right ventricle. Then what happens is the right ventricle squeezes and it sends blood out of the PA, which is the pulmonary artery. So what we do is you look here, there is the pulmonary artery. So what blood does, it comes out of the pulmonary artery. The L then stands for the lungs. So these are then the lungs here. So at the end of this, what we've got then is we've got the lungs. Pulmonary, it's deoxygenated blood. It's the only artery that has deoxygenated blood in it. Once it gets to the lungs, it then goes down the pulmonary vein, which is the only vein that has oxygenated blood. So if you find the pulmonary vein here, so it goes from the lungs into the pulmonary vein. Okay, there's a couple of them, because obviously there's two lungs. Right, and when it goes down the pulmonary vein, it goes into the left atrium, which is then the left atrium. We then go through the valve, which is here, into the left ventricle. This is then kind of the powerhouse of the actual heart itself. So you've got your blood now here inside the left atrium, left ventricle really, rather. Okay, and what that does, it's the biggest part of the heart and it's ready to squeeze it all the way around the body. And it does that going down the aorta, which is the largest artery. So if you look here, what it does, it goes up behind there, through this one here to the aorta. And then the aorta then distributes it down lots and lots of other arteries, okay, to distribute it all the way around the body. Okay, so here, the aorta takes it to the body. That's then when the oxygen gets deposited and carbon dioxide then gets picked up. So there's the aorta to the body. Now, what we're going to do then is, when the blood then starts to come back, it kind of collects in the vena cava, which is the VC one. So it goes up to the body, and then what it does, it goes then back down the vena cava. And there's lots and lots of veins that then connect into the vena cava. Okay, the blood goes into the vena cava, and then it reappears in the right atrium. And then what happens is the whole circulatory system then continues. So this is then my diagram. Right atria, let's just change the colour. Right atria, valve, right ventricle, pulmonary artery, lungs, picks up oxygen, pulmonary vein, left atria, valve, left ventricle, aorta, largest artery, body, gets rid of the oxygen, picks up carbon dioxide, vena cava, back to the start again. Right, now I talked a little bit about arteries and veins. Okay, now a good question is comparing them, right? And the way you compare them is, so if they say in an exam, you've got arteries and veins, compare arteries and veins, right? What you would do is you would say arteries have no valves, veins have valves. Okay, so what I've done here is I'll just go through arteries first. So arteries have no valves. So what that means is kind of like little pocket valves. It goes away from the heart. I like to say AOA, arteries away. It has a narrow lumen, which is kind of like the actual hole in the middle. It's under high pressure because it's straight from the heart. It's got thick muscular walls. Okay, you can see kind of thick muscular walls here. And it's mainly oxygenated. Okay, so if you remember, the only deoxygenated one is the pulmonary artery, because that is the one that goes from the heart to the lungs. Veins have got valves. Okay, what they are is little pocket valves to stop the backflow of blood towards the heart. Large lumen. Okay, you can see that here. Large lumen. Thin muscular walls, mainly deoxygenated. And the only one that is deoxygenated, or oxygenated rather, is the pulmonary vein. Okay, now they're great as comparisons. So what I would do is I'd look at the picture, right, and the picture kind of tells you a lot of the answers anyway. Capillaries are tiny, right? Probably if I was going to draw one, I'd probably do it like a little dot like that rather than something that looks like that, right? They have no valves, they're way small, they're under very low pressure. If you think about it in your fingertip, it's full of capillaries. If it was under high pressure, it would squirt out, so obviously it doesn't. It's one cell thick, and that is then to allow gas exchange and any other exchanges that are then going on. Blood. Okay, so if we're then going to start talking about blood, blood consists of 4 different parts. We've got plasma, we've got red blood cells, white blood cells, and we've got platelets. Okay, now what I've done on the side here is there was an exam question that came up about a year ago, and it showed a picture just like this. Right, and what you just got to do is you've got to get a bit of an idea about kind of the sizes of different parts of the actual blood itself. Red blood cells, they look like a donut, and they have no nucleus, so they'll tend to be the biggest, right, that are in there, and it'll also be the one that there's most of, because if you look at the percentages, 45% of the actual cells in there are red blood cells, or 45% of blood itself is red blood cells. Then you've got white blood cells, which have a huge nucleus in them. Okay, but there's not very many of them, right? So it's less than 1% of blood. We've then got platelets that are small little fragments of cells, so they're tiny, and again, there's not very many of them, there's less than 1%. And then the yellow liquid is then plasma. Okay, and that is kind of like the liquid that carries everything around. Now, if we go through each individual part, okay, if we start with plasma, it's a pale yellow fluid, right? And that's often yellow because what it's got urea in it. I'll just change the color because you can't see that. It's got urea in it, right? And urea is kind of one of the waste products of digestion, particularly proteins, right? And it's the yellow that's in your wee. Okay, so the yellow in there is often a urea, right, which is kind of a yellow waste product that your body is then going to filter out and eventually wee out. It also transports everything else. It transports carbon dioxide, hormones, waste, okay, anything that needs kind of delivering around the body is dissolved in the plasma. The only thing that isn't dissolved in, or one of the only things that isn't dissolved in the plasma is oxygen. So oxygen is in the red blood cells. Pretty much everything else is dissolved in the plasma. It has no nucleus, which then allows it to kind of bend and change shape, but it gives it a very short life, probably about 3 months. And it's full of a substance called hemoglobin, which has got iron in it, which gives it red colour. White blood cells is part of the immune system. So what it does, it produces antibodies, right, phagocytes and lymphocytes, which comes into a later topic. Platelets, all they do is they clot the blood. Okay, so we've talked about arteries and veins. Now what we're going to do is we're going to talk about coronary heart disease, sometimes written as CHD. Right, now on the right hand side here, what I've done a few diagrams to try and still illustrate it. So coronary heart disease, the cause of it is fatty substances build up, and that's called atherosclerosis. So that hopefully is your artery. This is somebody then who has then eaten too much fat, and they've got a build-up of fat in their arteries. And you can see that the yellow here is starting to block the artery itself. The effect of a build-up of fat in your arteries is the fact that oxygenated blood can't get around. And if it's in a cardiac muscle, so blood can't get to the cardiac muscle, then what eventually is going to happen is your cardiac muscle isn't going to have enough oxygen, and then you could potentially get coronary heart disease. Now, the way you can treat it, there's two main treatments. You've got stents and you've got statins. Statins is kind of like a food substance, right, that lowers harmful cholesterol. Statin is a more invasive treatment, which is where what you do is you get, it's kind of a bit like a tube. It goes in through the groin, right, up through a vein in the groin, right to where the actual issue is. And it's like a tube here that goes into the actual vein itself, and the yellow around the outside there is the fat. Then the stent expands, a bit like a balloon, and you can see it expanding there. Then eventually, when the balloon kind of pops or gets taken out, it kind of leaves a stent, which is a bit like chicken wire. Now, it's a bit like a sort of a metal mesh that's then left in place. And what that then has done is opened up the vein, and then it allows blood to flow properly. It's very clever. Okay, now just on to the intestine. Okay, we've talked about the intestine before, right? Now what we're going to do is we're going to look at a sort of gas exchange. The intestine, right, what it is, you would imagine it's a tube that just looks like that. Small intestine, if you cut all the way through it, as like an empty tube. But it doesn't. What it does, it has loads and loads and loads of little hairs sticking in. Those little hairs are called villi. Okay, so this diagram at the bottom right here, right, so what you've got here is you've got this one here, which is kind of like a zoom in of that initial one, and you can see lots and lots and lots of little hairs. Let's change the colour. Lots and lots and lots of little hairs sticking up. Each one of those little hairs looks like this. Okay, now the whole point in it is to improve digestion. And what they'll ask you is, how is the small intestine adapted to increase exchange? And there's three standard answers. The villi makes a large surface area, because if you think now, you can then get absorption all the way around that hair rather than just in an empty tube. It has a fantastic blood supply. The blood, you can see it all the way up the middle, the capillaries going up inside each individual villi. And you've also then got number 3 is a very thin wall, often 1 cell thick, right, to allow a very short diffusion path. Okay, so number 1 is a large surface area. Number 2, good blood supply, and that is then to keep the concentration gradients. And number 3 is then a thin wall, so there's a short diffusion path. Now, on to breathing, right? We'll go through the respiratory system. Okay, firstly what we'll do is we'll just go through the structure. All right, so you obviously breathe through your mouth. The air goes down your trachea, okay, which runs right next to your esophagus, down your neck. It goes into the bronchus, which is kind of where it divides. It then goes into the bronchioles. And then when it goes into the bronchioles, it then goes to an alveoli. Okay, and then the whole kind of thing itself. It's a bit like a tree upside down, really. So what you got is you kind of got that there is your trachea. It then branches off the first time, which is into your bronchus. Then it kind of separates again into kind of little twigs, which is like the bronchioles. And then at the end of each individual little twig, what you've got your leaves, which is a bit like the alveoli. And I always think that the alveoli look kind of like a bunch of grapes. Okay, so you've then got your alveoli, oxygen goes in, carbon dioxide comes out, and what you have is you have a little kind of vein, or capillary around the outside of each alveoli for gas exchange. Right, these are then the jobs of each part. So what we've got is we've got trachea. Okay, it takes air from the lungs, and what it's got rings of cartilage to stop it collapsing. We've got the bronchus, which is kind of the dividing point. We've then got the bronchioles, right, which then split into many, many different pathways to get air to the alveoli. Alveoli is then the site of gas exchange, and capillaries, there's loads and loads of them, is to then take away the oxygen and to deposit into the carbon dioxide. Now, this is then kind of a little bit more on the way that it works. Okay, so the blood comes down there, right, and what it does is carbon dioxide diffuses out. Okay, because that is then your waste product. And as the carbon dioxide diffuses out, oxygen diffuses in. And just like it was with the villi, there's three answers if they start asking about exchange surfaces. Number one, the alveoli gives it a large surface area. Number two, a good blood supply to maintain concentration gradients. And number three, thin wall for a short diffusion path. Okay, so as soon as they say the words gas exchange, right, or exchange of any sort, what you've got is you've got your three answers. Now, how do we breathe? Okay, so what we've got is we've got lung 1 and lung 2. Not that it makes any difference. Breathing is all about this thing here at the bottom here, the blue thing. That's your diaphragm. Okay, diaphragm. Okay, is your diaphragm. To breathe in, what you do is your diaphragm goes down. And what that does is it reduces pressure in your thorax, okay, which is kind of the bit above your diaphragm up to your neck. And if pressure is decreased in the thorax, what happens is air is then sucked in. So the lungs themselves have no muscular kind of content at all, right? So the thorax, when the pressure decreases in it because the diaphragm's gone down, it sucks air in. And then to breathe out, what happens is your diaphragm goes up, and then that increases the pressure in the thorax. So it increases the pressure, and air then gets forced out of the trachea. Right, non-communicable diseases. Okay, we talked a little bit before about heart disease. Now we're just going to talk about some others. NCDs refer to a group of conditions that are not caused by an infection. All right, so basically what a non-communicable disease is a disease that somebody can catch or has but cannot be passed on to somebody else. Most common kind of example of that is cancer. Okay, horrible word. And what cancer is, it's a result of where the DNA changes that can lead to uncontrolled growth and division. So in a nutshell, what happens is one cell, its DNA changes, it has a mutation, and what it does, it becomes cancerous, and then the growth of that cell itself is uncontrolled, and it just gets bigger and bigger and bigger. There are two types of cancer. The first one is a benign tumor, right, which, you know, what it tends to be like a tumor that's kind of in a bag. Right, so it's got kind of like a membrane around the outside of it. A malignant tumor is then something that isn't in a bag, right, or a membrane. And what it can kind of do, it kind of grows roots out. They look a little bit like roots, and it invades the tissues around it, and it can then spread to different parts of the body. And if it does that, then that is then a secondary tumor, which is then real trouble. the plant. Okay, so now what we're going to do is we're just going to talk about structure of the leaf. Okay, I'm just going to kind of go through the structure top to bottom, and then we'll talk about the roles of each one. At the top, we've got the cuticle, which I will often call a waxy cuticle. Then what you got is kind of got the upper epidermis, a bit like your skin. Then we've got the palisade cells. In there, and often what they won't actually put this on the diagram itself, is your xylem and your phloem. The next layer down is then the spongy mesophyll. I'll call it spongy, right, because it just helps with the spelling. And what you'll notice with the spongy mesophyll is there's lots of gaps, where on the palisade cells there isn't. We've then got the lower epidermis, which is kind of like the skin of the bottom of the leaf. We've then got a stoma, and on either side of the stoma we've then got guard cells. And then the very bottom is we've got a cuticle, which again is a waxy layer. Okay, now what I'm going to do now is I'm just going to go through the kind of the roles of each individual part of the leaf. Waxy cuticle prevents water loss. You'd imagine that a waxy layer on the outside of a leaf prevents water getting in, but it isn't, right? Its main role is to prevent water loss, right? If you think about a cactus, right? A cactus has a very, very thick waxy cuticle, and that is to prevent water loss. The upper epidermis, right? What that does, it's a very thin layer, right? And it allows light to get through, and it's also a bit like for protection. Okay, each of the green dots, okay, on there are your chloroplasts. Then what we get to the palisade mesophyll cells. They contain the most chloroplasts, and they are stacked vertically. Okay, makes sense really in the fact that these cells here, the palisade cells, have the most chloroplasts because they are the one that are directly under the sunlight. They are facing the sunlight. And they're also stacked in, so you can get as many in as you possibly can, which is clever. The spongy mesophyll, okay, has approximately 50% air spaces. That is then to allow the oxygen and the carbon dioxide to move and diffuse throughout the actual leaf itself. Okay, I'll talk about that a little bit more in a minute. The guard cells and the stomata come together. What the guard cells do is they open and close the stomata, right? And they do that by absorbing or releasing water, which makes them swell or reduce in size, right, to open and close the stomata. Okay, and the way that it works is when the stomata's open, we've got CO2. The stomata opens and the carbon dioxide goes in through the stomata, and it goes through the air spaces in the spongy mesophyll, and it gets to the palisade cells. Here in the palisade cells, you've got your light. So therefore you've got your light, you've got the carbon dioxide, you've got water that's been delivered by the xylem, and then you can do photosynthesis. When photosynthesis has then happened, oxygen comes out of the palisade cells, it goes through the gaps, and then it comes back out of the stomata. And that is then all done by diffusion. Right, transpiration and translocation. Okay, so we've mentioned very, very briefly before about xylem and phloem. Transpiration is all to do with the cells for xylem, and what xylem does, it then transports water, minerals and ions, right, from the soil to the leaves, flowers and fruits. Okay. I sometimes think about it as perspiration. Perspiration is human sweating. In a way, transpiration is a bit like water, plant sweating, but what it's doing, it's releasing water from the leaves. Translocation is moving of sucrose and amino acids from the leaves to anywhere in the plant at all. That is then done by phloem. Now, factors affecting transpiration. So remember, transpiration is a loss of water pretty much from the leaf. Okay, the factors that can affect it are temperature, wind velocity, and humidity. Okay, so again, if you think about it, this is water. So with the temperature one, okay, it kind of makes sense really. As the temperature gets hotter, the amount of transpiration increases until it gets to a certain point. With wind velocity, transpiration keeps on increasing as the wind gets faster and faster. Right, if you think about it, it's kind of blowing the water away from the top of the leaf. And humidity is transpiration happens a lot at low humidity. That humidity is the amount of water that is naturally in the air. And as the humidity increases, what happens is transpiration reduces until it gets to the point where the actual water concentration is greater outside of the leaf. And then transpiration doesn't happen anymore, right, because of the humidity. Now, measuring transpiration, right, I love this kind of little sort of diagram here, right? So what we've got is we've got a plant at the end here that is transpiring. It's got a cut root, and what happens then is you've got loads of water in here. The most important part of this then is how do you actually measure the transpiration? And it's all about this air bubble here. That little tiny air bubble there, right, it moves when the plant transpires. So as the plant transpires, what the little air bubble is, it moves along this scale. And what you can do, you can measure the amount of transpiration or amount of water lost based on the distance that that little air bubble moves. Now, the biggest problem that you ever do if you actually do this as a practical, right, is how do you get the air bubble back again? And what you do is you use this reservoir at the top here, and if you open up this little tap, water goes back down in here, and then it forces the air bubble back again, right? But it's a good way of measuring transpiration. Okay, now what we've got here is these are then the 5 questions that you need to write down in your exercise book. Number 1 is what is transpiration and what are the 3 main factors that affect it? So I want a definition and information about what transpiration is, and then list the 3 main factors that affect it. Okay, don't forget you may well get one of the graphs in your exam. Number 2, compare malignant and benign tumours. So compare means this does this, this does this. A malignant tumour has this, a benign tumour has this. Number 3, how are lungs adapted as a surface, exchange surface? 1, 2, 3. Question number 4, what are the 2 main raised coronary heart diseases treated? 1, 2. Number 5, list 3 roles of the stomach. 1, 2, 3. And remember, number 3 is the most important. Right, that is then the organisation topic. It's a very, very big topic. And you need to know all the different facts and all the different parts of information about it.